Okay, right. We are rolling. You are themselves just a subgroup of a larger group called the Alveolata. And the Alveolata are a subgroup of an even larger group called SAR. SAR is straminopiles, alveolates, and rhizaria. There aren't that many parasites that are straminopiles or alveolates or rhizaria, sorry. There aren't many straminopiles or rhizarians that are animal parasites, although there are a few. But alveolata includes the Apicomplexa, which is one of the really big parasite groups. But there are some other parasites in the rest of the alveolata that we ought to look at before moving on to the next major group that includes lots of parasites. Alveolata is Apicomplexa plus a group called the Ciliophora plus a group called the Dinoflagellata. So let's look at those real quick. Ciliophora are usually called ciliates. The vast majority of ciliates are free-living, and they include critters you've probably seen before. You might have looked at paramecium in intro courses. In cell biology, I believe you do a phagocytosis lab with a ciliate called tetrahymena. There might be others that you'll see. You'll spot them in just about any freshwater sample that you could take around here from lakes or ponds. There's plenty in the oceans. They're all over the place. They're a very diverse and successful group. What makes them a group is they're covered in cilia. Eukaryotic cilia and flagella are basically the same thing. The difference is whether they're long, they're flagella. If they're short, they're cilia. But the cilia here are arranged in rows. They're called kinetes. And the cilia have this root structures. They're rooted with this arrangement of microtubules called kinetids. And the cilia beat in what's called metachronal waves. You've seen metachronal waves if you've ever seen those electric signs that have lights flashing on and off so that it looks like there's maybe a wave of light going around. But of course, nothing is really moving. What's happening is the lights are flashing on and off. Or better yet, a great example of a metachronal wave is the people doing the wave at like a football game. Right? People aren't really moving. They're just standing up and sitting down, but you can see a wave of, people standing up passing all the way around the stadium or all the way down the bleachers or whatever it is. That's a metachronal wave. There's nothing that's actually changing position, but a wave of movement is passing in a particular direction. Like this, if you can see it. There's a metachronal wave for you. Ciliates usually have a defined mouth, quote, called a cytostome, a particular spot where food particles get taken in. Their mouth may be in a groove. There's often a complex cytoskeleton surrounding it. There may be specialized cilia around it. There's little things that can shoot out under the surface called extrasomes. This gets into sexual reproduction, and I don't think I'm going to talk about that much. But they can reproduce sexually or asexually, and each cell has a typical eukaryote diploid nucleus called a micronucleus, and then this weird bag of DNA fragments called a macronucleus. That nuclear dichotomy is fundamental to ciliates, but I won't say too much about it here. If you take or have taken invertebrate zoology with me, you'll hear more than anyone really should ever have to. Okay, there's a typical one that's Tetrahymena. This is one that you've probably seen before or will see if you've taken cell. You can see the cilia covering the entire body, and you can see that they're arranged in rows. The cytostome is at the top and a bit to the right, and you can't clearly see it, but there's some cilia that are spaced very densely around the cytostome. And those cilia are so close together that they essentially beat as one, and they're called membranelles. They're not truly a membrane, but they're packed so tightly together and they beat in sync that they essentially act like a membrane, a thin extension flapping back and forth as a unit. And tetrahymena means four membranes because this particular genus has four membranes surrounding the cytostome. Right. Now remember, alveolates are called that because under the plasma membrane, they have flat sacs made of membrane called the alveoli. The best thing I can think of comparing this to is quilting. Imagine you have a quilt and then a sheet on top of the quilt. Well, the sheet on top would be the plasma membrane. Underneath the sheet, you've got cloth that's stitched to make these flattened compartments. And in a typical quilt, the compartments are stuffed with soft stuffing, quilt batting, they call it. So that's the best metaphor I can come up with. This happens to be my favorite ciliate of all. It's called euplotes, and it's been stained with fluorescent antibodies to specialized proteins that are in the alveoli, and you can see this beautifully. The front end of the cell just looks like a mosaic. Each one of those little polygonal tiles is a single alveolus, and you can see how they're packed together very tightly and they fit together very tightly. All of that is just underneath the outermost plasma membrane. And remember, the apicomplexans have this too. Dinoflagellates, which is the third group of alveolates that we'll mention today, has this too. Okay, right. What about the parasites? There aren't that many, at least in, there's really only one that's much of a parasite in humans. And it looks like this. It's called Balantidium coli. It is often asymptomatic. It can cause ulcers similar to entamoeba ulcers in the gut. I'm not sure that this has been studied much. It doesn't seem to be particularly common. It forms infective cysts in the large intestine and is transmitted by the fecal oral route. So there is one ciliate that parasitizes humans and sometimes causes disease. There are a number of ciliates, by the way, that live in cow stomachs, particularly in the first of the four stomach chambers that a cow has, the rumen. Those are actually mutualistic. They help in the digestion of food. And other ruminants have in there as well. A number of ciliates that are commensal with cows, and as far as I know, all animals that chew the cud. They're not parasites, so I'm not going to say much about them. The only other parasitic ciliate that you might ever be likely to encounter is one that attacks fish. And this is what it looks like. This is Ichthyophthirus multifilus. It causes a disease that's commonly just called ick in freshwater fish, in particular in aquariums. If any of you have ever kept a freshwater tank, this is exactly what you don't want to see on your tropical fish, being covered in white spots like this. That looks like they've been salted. Each one of those white spots is a cyst on the fish's skin, caused by infection with Ichthyophthirus ciliates. And if this happens, you have to add medication to the water, and if you don't, it can go right through your tank. So anybody who's ever kept or might keep a freshwater tank might encounter this at some point, or if you've ever worked in a pet store, you might see this. That's what it looks like. It forms those cysts, those white specks in the skin of an infected fish. When it's mature, they drop off and they form another type of cyst on the bottom and divide repeatedly. And multiple infective cells come bursting out of those cysts and then go on to invade new hosts. That's all I'm going to say about it. That's one of the few ciliates that significantly parasitizes vertebrates. If any of you ever work in an aquarium or in the pet trade or just keep a tank, it might be something you'll encounter. The third group of Apicomplexa, talked about ciliates. The third, about half of all species are photosympathetic. Evolutionarily, they've actually acquired their chloroplast by stealing them from a variety of sources. Many of them have chloroplasts that used to belong to green algae. It's called kleptoplasty. There's a number of cases where one protist group has taken chloroplasts from another protist group, probably by engulfing them and then retaining the chloroplast in their own cell. It's kind of weird, but they can do that. Most dinoflagellates can also feed. They've got a pair of flagella, one of which pushes from behind and the other one of which is wrapped around the cell. When they both beat, the one that's wrapped around the cell causes the cell to spin as it swims forward. And dinoflagellate, in Greek, dinos can mean terrifying. That's what it means in dinosaur, terrifying lizard. But there's a similar root that means whirl. So dinoflagellate does not mean terrifying flagellates, although as you'll see, they can be terrifying. Dinoflagellate means whirling flagellates, spinning flagellates, because of that transverse flagella. Membranes in dinoflagellates are filled with cellulose, and they create this armor-like coating just under the plasma membrane. You'll see that in the next slide. It's called a theca. There are a few dinoflagellates that don't have those cellulose deposits. They're called naked dinoflagellates. There's a few genera that are very important as mutualistic symbionts. Reef-building corals have lots of symbiotic dinoflagellates in their tissues and depend on them to survive. If corals kick their symbionts out, it's called coral bleaching, and a reef that does that is in real trouble. This is something that we've seen a lot of in recent years. In the oceans, you can also get these dinoflagellate population explosions. This causes what's called a red tide, and this can be dangerous because some species release very large amounts of toxins. If you eat seafood that's been feeding on toxic dinoflagellates at certain times of the year, you can get quite sick. At some times of the year, if you eat oysters or something else that filter feeds, you may be eating oysters that have a lot of dinoflagellate toxin, and you can get this thing called paralytic shellfish poisoning, which is not fun. That's a scanning electron micrograph of a fairly typical marine dinoflagellate. You'll notice that it looks like it's made up of plates butting together, making these kind of irregular polygon shapes. That's the theca that you're seeing. And remember that these fecal plates form inside the alveoli. And this should remind you of that euplotes ciliate picture that we saw earlier. This one, in fact, that's the ciliate. And you can see how those alveoli fit together very tightly. Well, here on the dinoflagellate, you can see the same basic structure. And you'll notice there's a groove running right around the equator of the cell. And if this was living, one flagellum would be streaming out below, and the other one would wrap around the cell like a belt. There's a red tide. Notice there's a bunch of dead fish floating in the water. This is caused by a bloom of a particular dinoflagellate species, Karenia brevis. Many dinoflagellates produce toxins. And again, if dinoflagellates are fed upon by fish or shellfish, the toxins can build up and can cause serious illness in humans that eat the shellfish, like paralytic shellfish poisoning and an equally nasty disease called ciguatera. This is one of the reasons why the rule of thumb is you should only eat oysters in months with an R. Do you guys know that? You shouldn't eat oysters in May, June, July, or August. And one of the reasons is that's when they're spawning and they've used up all of their metabolic energy to make eggs and sperm, so they're not very tasty. The other reason is this is the season when you're at greatest risk of a dinoflagellate bloom making your oysters toxic. So only eat oysters in months that have an R in them. Now, you know, don't ever let it be said I don't teach useful advice. This is one dinoflagellate that has been implicated in directly attacking fish tissues, causing outright disease in fish. It's been called the cell from hell. Researchers that have come into contact with the toxin that Pfisteria makes have reported physical and mental symptoms, things like memory loss. So the toxin here is supposed to be really, really bad. By the way, that thing that looks like a spring that's kind of wrapping around this cell, that's that transverse flagellum I was talking about. And these are lesions on fish that have Pfisteria growing in them. We now think, though, that Pfisteria isn't a parasite, it's an opportunist. It actually attacks lesions on fish, but the lesions, the wounds, were made by a different parasite, a water mold. A water mold called Aphanomyces. We talked about water molds a way back. They look vaguely like fungi, but technically they are not. They're in that Stromenopiles group. So it seems that Aphanomyces makes the initial wound, and then Pfisteria can colonize the wound. We say that's an opportunistic infection. Pfisteria has been claimed to have an absolutely ridiculously complicated life cycle, going through a whole bunch of stages in which it can look like all kinds of different things. I think we now think that the life cycle is not nearly that complicated, but there still is work that needs to be done on Pfisteria. One parasite that is economically important is a little thing called Perkinsus marinus. And this is now classified not quite in the dinoflagellae, but closely related to it. It's a group called the Perkinsozoa. And it matters from the human perspective because it's a major pathogen of oysters. I don't know how you folks personally feel about oysters, but they are a pretty major seafood product. Most oysters these days are farmed, and that's a billion-dollar industry. So anything that affects oysters has the potential to have some pretty bad economic fallout. On the left, you can see what these look like when they've been stained. The cells are, less than 5 microns long. They're quite small. On the right, you've got a transmission electron microscope of a cross section through them. They've got 2 flagella labeled F in the picture on the right. One of them is that thing that's trailing down and to the left. The other is that round dot. That's a flagellum that's been sliced clean through. That's a cross section of a flagellum. So they're these dinky little swimmy things with 2 flagellates. The stage that you're seeing, called a zoospore, is ingested by an oyster, because oysters just filter feed and slurp up anything. Within the oyster, the zoospore transforms into a trophozoite stage. You've seen that word before. The trophozoites will invade cells in most of the oyster's tissues, divide, and release up to 16 new trophozoites from every cell. Some of those might reinfect the oyster. Others will leave the oyster and transform into new zoospores, and round goes the circle of life. On the left are normal oysters, quite edible, quite tasty with the right mixture of ketchup and horseradish sauce. On the right, you have oysters infected by Purkinje's, and you can see they're not nearly as plump. They look almost withered. So the effect, oysters' tissues waste away. The oysters can't reproduce very well, and you also get pus pockets in the tissues. And I just heard something go ding-a-ling, so give me just a moment. The disease is called dermo because Purkinje's used to be classified as something called dermocystidium. Okay, sorry, it was just somebody typing in their names. So yeah, you got normal oysters there on the left and an infected one on the right. Okay, that's all we're going to say about Alveolata. So that's Apicomplexa, which is a big group of parasites, and then there's a few parasitic groups in the Ciliophora and the Dinoflagellata. The Alveolata are a subgroup of a super duper group called SAR. The other super duper group that's got lots of parasites in it is called Excavata. Excavata, most of these have flagella, right? Long hair-like organelles that they'll use to swim. They get their name because many of them have a specialized food groove in the cell. Not all of them do, however, but many of them do. Many of them have lost their mitochondria. They retain DNA sequences that used to belong to their mitochondria. One of the things that's happened in the 2 billion years or so of the mitochondrial symbiosis is a lot of mitochondrial genes have gotten transferred to the host nucleus. So that's kind of weird. You've got genes in your genome that used to belong to bacteria. You've been genetically engineered. Ha ha ha. That sort of thing happens a lot more often than we used to think. And so excavates, many of them don't have mitochondria anymore, but they've got DNA sequences that suggest that they used to. So they've lost them secondarily. This is also true, by the way, of entamoeba, the parasitic amoebas that we looked at. Some excavates still have organelles that are probably adapted, modified mitochondria, including some ATP-generating organelles called hydrogenosomes that ferment pyruvate. Okay, Plenty of excavates are free living. You could see some in just about any old pond water sample, but there's a lot of symbionts and a number of important pathogens in the Excavata. So this is the second really big parasitic protist group that we're going to focus on. There's two major clades, and I'm not going to say much about how the basis of this classification because it tends to involve DNA sequences and some ultrastructural things. Okay, the Discoba includes Euglenozoa. That includes some free-living protists that you might conceivably have seen before, like Euglena, but it also includes a very big group of parasites called the trypanosomes, which include the causative agents of African sleeping sickness and also of Chagas disease. Second group in the Discoba, the Heterolobosia, includes the amoeboflagellates, which include the infamous but mercifully very rare brain-eating amoebas, which we'll talk about later. I've always loved brain-eating amoebas. I did my undergraduate honors thesis on a relative of the brain-eating amoebas. So, sorry, kind of a fanboy here. Okay, the second group, Metamonada, includes a lot of insect symbionts. I might actually do this as a demonstration for you guys. Termites are noted for having lots of protist symbionts in their guts. And some of the 1440 instructors, not all, used to, I don't know if they do it today, but they used to have their termites squish termite guts because there's all kinds of crazy things growing in termite guts. It's pretty awesome, almost as cool as roaches. Most of those symbionts are in the Preaxostyla and the Parabasalia. Some of the little flagellated protists that you, at least one of you saw some in your cockroach guts, they're also in this group. And then finally, a metamonad subgroup called the Fornicata includes a major parasite called Giardia, the causative agent of what they call hiker's diarrhea or beaver fever. Beaver with a V. Beaver fever with a B is something very different. Right. Okay, so it's going to take probably most of this week to cover all of those. That first group I mentioned, the Euglenozoa, includes free-living excavates called euglenids, whose ancestors picked up chloroplasts by engulfing a green alga. And these are pretty common in fresh water. They swim actively. They're also a deep grassy green. You can see Euglena, for example, over there on the left. On the right is another flagellate that you commonly see in pond water. This one happens to be called Paranema. It doesn't have chloroplasts, but other than that, it's very closely related to Euglena. It's in this group called the Euglenozoa. And again, I won't say much about them, but these are critters that you'll see if you ever spend time staring down the barrel of a microscope at pond water. They are very, very common. The closest relative of the euglenids within the Euglenozoa are the kinetoplastids. And there are some of these that are free-living as well. They tend to be kind of smallish, colorless little flagellates that you can sometimes pick up in pond water and places like that. But kinetoplastids also include quite a lot of parasites. On the left, trypanosoma, the causative agent of African sleeping sickness and a disease endemic to South America called Chagas disease. On the right, leishmania, which causes various diseases in the developing world, notably a very severe disease in Central Asia called Kala Azar, which I'm told is Turkish or Persian for the black disease, frequently lethal without treatment. So here's a close-up of some trypanosoma, and the cells are kind of shaped like crescents or shaped like bananas, I guess. And if you look carefully at each one, each cell has a nucleus. That's the larger of the dark spots that you see. Each cell also has a flagellum, and in trypanosoma, the flagellum kind of attaches to the side of the cell and then extends along the length of the cell and is actually tethered in place by a kind of web, a kind of fin made up of a fold of the plasma membrane. So instead of the flagellum just sticking freely out from the cell and waving in the breeze, the flagellum is tethered to the cell by this fold of the plasma membrane and forms this undulating fin, this fin that bends into S curves and enables the cell to swim. Right where the flagellum attaches to the cell body, there's a darker spot, smaller than the actual nucleus. The nucleus is more or less in the middle of the cell. The darker spot is right where the flagellum emerges. The darker spot is called the kinetoplast. That's why trypanosoma and others are classified in a group called the kinetoplast. There's a diagram of what trypanosoma looks like. It emerges towards to the right, and you can see it's labeled attached flagellum, and it extends to the left, and it's tethered to the rest of the cell by the undulating membrane. And then it kind of goes free way out there towards the end. You can see the nucleus with the nucleolus more or less in the middle. And right where the flagellum attaches, there's a little granule, the blepharoplast. And then just in front of that, you have a dark spot called the kinetoplast. Turns out the kinetoplast is a big blob of mitochondrial DNA. And I'm not sure I understand why it does this, but the kinetoplast is made up of a whole bunch of circular pieces of mitochondrial DNA that are all chained together, right? Making the kind of thing that you might have made once if you ever had about 100 key rings and an awful lot of time on your hands. There's dozens of larger circles of mitochondrial DNA called maxicircles, interlocked with thousands of smaller bits of DNA called mini circles. And the whole thing is big enough that you can see it under the light microscope. So, I mean, I know you've heard that mitochondria are descended from bacteria that penetrated an early DNA. This is one of the few cases where you can actually see mitochondrial DNA. That little dark staining, the small nucleus, is the DNA of a big mitochondrion. So when we talk about trypanosomatids, we often have to discuss the different shapes that they're capable of taking. And one species may undergo several changes of shape. Leishmania, for example, alternates in its life cycle between promastigotes and amastigotes. We'll talk about that when we get there. Amastigotes don't have a flagellum at all, or if they do, maybe a tiny little stump of one, but essentially you can't see it at all. That's what you see on the left. Promastigotes, the flagellum is at the anterior posterior end, right? So pulled by its flagellum, pulled from ahead, not pushed from behind the way a sperm cell would be. Pistomastigote is embedded in a very deep pocket inside the cell. Epimastigotes, the flagellum kind of emerges from the middle of the cell and runs along the side. And then trypomastigotes are what we just saw, where the flagellum is attached to the length of the cell by an undulating membrane, a fold of the plasma membrane that makes what amounts to a fin. There are some that you can see in insects, Leptomonas and Herpetomonas. I used to do a lab where I'd go out and I'd catch either fruit flies or house flies. And if you can pull out the fly's foregut and salivary glands, roughly 10% of your average wild fruit fly population will have Herpetomonas living inside. And they're kind of neat to see if you can find them alive. They look like tiny little twitching carrots. I don't do that. I might have done it if we were doing the usual complement of labs, but this is one that I dropped because I found that only about 10% of your average wild population will have these. So if I'm able to bring in enough flies for every student to dissect one, I might get two that will see Herpetomonas. And that's neat, but I want to get labs ideally where more students can get positive results if I can manage it. So they're there, and some years we get to see them, but I'm not doing it this year. The whole life cycle is carried out in insects. Some of them seem to be pretty much commensal. Others can cause disease. There's actually and has actually adapted to live the plant's phloem, is disease of coconut palm coffee. So if you like coconut or you like coffee, this is something to affect you. What you're looking at, you see a scanning electron micrograph of the whole Ciliophora, and above it, you're looking at a cross flagellum, or from that flagellum pocket that you might be able to see. And way. But the ones we tend to be concerned about, one causes of humans, Trypanosoma brucei, causes African sleeping sickness. This affects not just humans, but animals as well. It's spread by the bite of an African fly called the tsetse fly, Glossina morsitans. They reproduce within the human bloodstream, are picked up by the bite of a tsetse fly. They reproduce within the fly and enter the fly's salivary glands and get injected when the tsetse fly bites another victim. There's indirect evidence that these go through a sexual stage within the fly, but we've never really caught them at it. We don't really know what that looks like, but we find evidence of genetic recombination in Trypanosoma taking place somewhere in the fly. So there are some superficial similarities with malaria. It's spread by blood-sucking flies, and it undergoes sexual reproduction within the fly, and it's strictly asexual, as far as we know, within humans. The natural host of Trypanosoma brucei are native African animals, antelopes and things like that, and they're only mildly affected by it. They have evolved a good deal of resistance. Cattle, however, suffer very severe disease, a disease called nagana. And most cattle can't be raised in areas where Trypanosoma brucei is endemic. The exceptions are cattle breeds like this. There's at least one of our students is a cattle aficionado, lives on a cattle ranch. So you might have some professional interest in these West African Ndama cattle. These have evidently evolved a great deal of resistance to Trypanosoma brucei. There's 3 subspecies. Trypanosoma brucei only infects cattle and large African mammals. It does not infect humans at all. If you can see, there's a black line on this map of Africa. To the left of that line, in Western Africa, you get Trypanosoma brucei gambiense, which is endemic to Western and Central Africa. This is the one that causes the vast majority of human cases. To the right of that line, Trypanosoma brucei rhodesiense infects only about 100 persons a year. It's still bad, but not as much of a threat as Trypanosoma brucei gambiense tends to be. So you get this by being bitten by a tsetse fly. The first sign of infection is an ulcer that develops around the site of the bite. It's called a canker. And when the parasites have been injected into your circulatory system, start getting symptoms. It comes and goes. Your spleen may enlarge, joint pain, itching, and these can affect you for about a year. If it's gambiense, if it's rhodesiense, it might be only a month. After this time, when the disease is present in your circulatory system and your lymphatic system, the parasites start entering your nervous system. They get into your cerebrospinal fluid. Once they're in there, they start causing neurological symptoms. Basically, every bad thing that can happen to the brain, they can cause it. So you could get hallucinations, confusion, memory loss, loss of speech, tremors, partial paralysis. Ultimately, if it's untreated, it ends in coma and ultimately death. One particular thing it will do is disrupt your sleep cycle. Infected people may start getting very drowsy during the day and then may not be able to sleep at night. This is the reason why it's called sleeping sickness. And here's a patient being treated for it. There are, fortunately, a number of drugs that are effective. Although if it's not caught early, you can end up with lasting neurological damage. Very rarely seen in this country. When you do get a case of it, it's generally brought back by a traveler, people who've been on safari or something like that, or I guess it's possible for immigrants to have it, but you won't see it often here. But it can be a public health problem in Africa. When we reconvene, I'll talk about another one that's a public health problem in South America, one that has also been spotted in North America as well. I refer, of course, to leishmania. Let me see. Do we have any questions? Okay, the chat says I was breaking up for some people. I'm sorry about that. I don't know what that is, but I'm going to go ahead and stop recording.